Growing up with food allergies, being a food allergy awareness advocate, studying and researching food allergies, and now working in the food hypersensitivity regulation field, and getting to interact with world-leading researchers and experts, I have developed a passion for understanding all aspects of food allergy and other food hypersensitivities. In this podcast, we will be taking a closer look at the science behind food hypersensitivity and dissect some of the misinformation we often read or hear about. We will also hear stories from people with lived experience. My aim is to create a scientifically curious community and to get you interested in the science behind food allergies and other food hypersensitivities. So in this episode, I'm going to be discussing all about the risk factors that increase the risk of developing food allergy. I just wanted to quickly mention that I might use some of the terminology that I used and explained in episode 2. So if you're interested, then I'd highly recommend listening to episode 2 before coming to listen to episode 3. So IgE-mediated food allergy is increasingly becoming a public health concern, with prevalence on the rise in the UK and really globally. And this is why it is so important to really understand what the risk factors are that contribute to food allergy development. IgE-mediated food allergy is a complex disease, and this just means that both genetic and environmental risk factors affect its development. IgE-mediated food allergy is also a polygenic disease, and this just means that it is influenced by more than one gene. Numerous risk factors have been proposed to contribute to allergy or sensitization status. And in this podcast, I will address a few of the most researched genetic and environmental risk factors that increase the risk of developing IgE-mediated food allergy. So how do we know that genes play a role in the development of food allergy? Well, we need to observe if a trait is inherited, so runs in families. And identical twin studies are really valuable because identical twins share 100% of their genes, whilst non-identical twins, just like siblings, on average share about 50% of their genes. If identical twins show more similarity on a given trait compared to non-identical twins, this provides evidence that genes are significantly influencing the trait. And previous family and twin studies have shown that IgE-mediated food allergy is highly heritable, meaning that an individual can inherit genetic markers that can predispose them to developing IgE-mediated food allergy. In a small twin study on peanut allergy, one study reported that the concordance rate of peanut allergy was 64.3% among identical twins and only 6.8% in non-identical twins. This study also estimated heritability of peanut allergy at 81.6%. This demonstrates the influence of genes have on IgE-mediated food allergy, as those with more similar genetic backgrounds have a greater chance of developing IgE-mediated food allergy. Now we know that genes play a role, let's dive a little bit deeper and investigate some of the most researched genes that have been found to influence food allergy and its development. Now, a gene is just a small sequence of DNA on a chromosome, and genes code for particular sequences of amino acids which can make up a specific protein. Within that sequence, you can have variations. And these variations in gene regions are really what we look for when investigating if a gene influences a trait. So genetic variation is very, very common in the general population, and often it contributes to the differences between populations and between people. It's really what makes us unique. However, these variations within our genes can also contribute in a small and additive manner to a complex disease such as food allergy, and so is incredibly useful to research. In my master's research project, I focused on looking at these variations, which increase the risk of IgE-mediated food allergy. I performed a systematic literature review where I essentially reviewed all of the primary literature out there that investigated a type of genetic variation called a single nucleotide polymorphism, SNP for short, to see if certain SNPs are associated to IgE-mediated food allergy. I created a database of 251 candidate SNPs in 46 gene regions, which were significantly associated to food allergy from 42 studies. I then used bioinformatic techniques to analyze these genes and SNPs, along with a further 19,039 SNPs, in a cohort of patients who were either sensitized to egg, peanut or milk, or inhalant sensitized to cat, dog, grass or dust mite, or both. I identified various variations in various gene regions that are associated to atopic sensitization status. Now let's investigate some of the genes that have been linked to food allergy. The most researched genetic region which has been shown to play a role in many atopic conditions such as asthma and IgE-mediated food allergy is the HLA region. The HLA gene region codes for the human leukocyte antigen complex, and this is a group of proteins that are involved in the regulation of the immune system. These proteins are found on the surface of cells and are responsible for identifying foreign invaders in the body, such as food proteins, and binding to them. And genetic variation has been found and reported in different parts of the HLA gene region, and this has been associated to food allergy. This is because these variations cause changes in the groove of the HLA protein. And because this protein is involved in immune regulation, changes to its structure can affect how it binds to allergens, and this can trigger an immune response. Variations in the HLA region can increase the risk of sensitization to certain food allergens. So the next gene I'm going to discuss is extremely important because of the dual allergen exposure hypothesis. This hypothesis was revolutionary in many ways, and numerous studies have found research evidence to support this hypothesis, and now genetic evidence backs it up. So what is the dual allergen hypothesis? Well, the dual allergen hypothesis states that exposure to food allergens through the skin can lead to allergy, whilst consumption of these foods at an early age may result in tolerance and reduce the infant's chance of developing food allergy. So, for example, if a baby has a disrupted skin barrier by having eczema, this could allow exposure to food proteins in the environment, such as peanut oil in creams or peanut residues on the table. And under the hypothesis, if these children avoid peanuts but are still exposed to them in the environment through their skin, they might be more likely to develop food allergy and specifically peanut allergy. And genetic variation within the skin barrier genes can be responsible for defects in the skin barrier. So it's really important to really understand what genetic variations in what genes can affect skin barrier function and potentially cause an increased risk of developing food allergy. The most researched gene is the FLG gene, and this has been investigated in many studies. The FLG gene codes for filaggrin, which is a structural protein involved in maintaining the skin's structural integrity. And mutations in the FLG region compromise the skin permeability, thus making someone more susceptible to developing IgE-mediated food allergy by enhancing allergen entry through the skin. One study found that a direct relationship is seen between having eczema and having FLG loss of function mutations. And as we discussed earlier, Having eczema can increase the risk of developing food allergy because it allows allergen entry through the skin through defects in the skin barrier. Now, there have been many genes studied which have a role in gene transcription, cell proliferation, cell migration, cell fate, cell adhesion, apoptosis, and various epigenetic controls that have also been linked to increasing the risk of IgE-mediated food allergy. However, I will not really be discussing this in this episode. Now let's move on to some of the environmental risk factors that increase the risk of a child developing food allergy. And in this section, I will discuss findings from studies which have investigated early introduction of food into the infant's diet, moisturization frequency, and vitamin D status. Now, I just wanted to clarify that I am only sharing study findings with you, and I'm not in any way offering advice on how to reduce the risk of developing food allergy in infants. It is really important that until the results are translated into government guidance, that parents continue to follow existing government advice and discuss any questions or concerns they have with their treating doctor or healthcare visitor or allergy specialist. Now, there's a misunderstanding that by completely avoiding and not introducing an infant to a food item, that they won't develop food allergy. And we've seen in various studies that this proves to be the opposite. So the Learning Early About Peanut Allergy study, the LEAP study for short, was a groundbreaking study which investigated whether early introduction of peanuts into a child's diet reduces the risk of food allergy development. This study identified 640 infants between the ages of 4 to 11 months with either severe eczema or egg allergy or both. These infants were considered at a very high risk of developing peanut allergy and were randomly assigned into two groups. One group consumed peanuts and one group avoided the consumption of peanuts. Infants assigned to the group which consumed peanuts were given at least 1 1/4 teaspoon of either smooth peanut butter or Bambay, a peanut-containing snack consumed regularly by children in Israel. The study showed that early consumption actually benefited both groups in reducing the prevalence of peanut allergy, and this again supports the dual allergy hypothesis. Another revolutionary study was the Inquiring About Tolerance study, the EAT study for short. The EAT study tested the hypothesis that early introduction of certain allergenic foods into the infant's diet may stop them developing allergy to those foods. The EAT study followed the same basic hypothesis that the LEAP study followed, but whereas the LEAP study focused on peanut allergy in high-risk infants and children, the EAT study looked at the introduction of a wider range of foods in the general population. The EAT study recruited 1,303 mothers and their infants, and all mothers on the study were to breastfeed exclusively until 3 months of age, at which point they were randomly split into two groups. One group followed the standard UK government advice and were asked to exclusively breastfeed for around 6 months of age, after which introduction of allergenic food was a matter of parental choice. The second group was asked to introduce 6 allergenic foods from the age of 3 months, alongside continued breastfeeding, with the help of a dietitian and support from the study team. And the EAT study found that introducing the allergenic food into the infant diet from 3 months old may actually be more effective in preventing food allergy development. And so they found that those children who, had these allergens introduced into their diet at a much earlier age had a much more reduced rate of developing food allergy, so they had much lower levels of food allergy development. And so actually introducing allergenic foods into the infant diet from 3 months may be effective in food allergy prevention when sufficient amounts of allergenic foods are consumed. So overall, the early introduction of allergenic foods may reduce the risk of food allergy development. Now let's move on to moisturization. So it has been demonstrated by multiple studies that a disrupted skin barrier and an inflamed skin barrier is the root cause of the development of food allergy. Now, could it be that if we prevent the damage to the skin barrier that we can reduce the risk of developing IgE-mediated food allergy? And if this is the case, how do we prevent damage to the skin barrier? Well, the Barrier Enhancement for Eczema Prevention study, the BEEP study for short, investigated the hypothesis that intensive moisturization from birth would prevent the development of eczema by enhancing the skin barrier. However, they actually found that moisturization intervention did not prevent eczema, and they even found that there was a non-significant increase in food sensitization and IgE-mediated food allergy at 2 years of age in some infants who were moisturizing more. Now, remember when I discussed the EAT study? Well, families involved in the EAT study completed questionnaires that included questions about frequency and type of moisturizer applied, and the use of corticosteroids, creams, and also parental reports of dry skin or eczema. And infants were also examined for visible eczema at enrollment visits. A relationship was observed between parental reported moisturization frequency at 3 months of age and the subsequent development of food allergy. And this relationship was present in both infants with and without eczema. Now, these findings are just huge. These findings actually support the notion that regular application of moisturizer to the skin of young infants may actually promote the development of IgE-mediated food allergy. Now, there are 2 possible explanations for these findings. Firstly, that moisturizers might actually facilitate the passage of food allergens across the skin barrier, or that moisturizers might actually damage the skin barrier and allow the passage of food allergens and aeroallergens. These findings are potentially of great significance, and further research is required to really understand the mechanisms that are underlying this, and also to really understand whether moisturizers facilitate food and aeroallergen uptake, and if moisturizers can become contaminated with these allergens from the hands or from the environment, and even if it is important, in fact, and whether the effects are limited to certain types of moisturizers or to specific susceptible individuals. So now we understand that frequent moisturization in infancy may actually increase the risk of development of IgE-mediated food allergy. Let's now move on to the last and final risk factor that I wanted to talk about today. And this is vitamin D. Now, in the past decade, there have been a rise in the number of studies investigating the role of vitamin D in the prevention of IgE-mediated food allergy. However, this remains controversial. Almost all cells of the adaptive immune system express the vitamin D receptor, making them also capable of being vitamin D responsive. Therefore, it is plausible that vitamin D might be required for regulating immunological mechanisms that are important in preventing food allergy and establishing oral tolerance. Many studies report an association between sunlight, the season of birth, and latitude as increasing the risk factors of IgE-mediated food allergy. The Australian Health Nut study shows that children born in summer have a 55% lower chance of developing IgE-mediated food allergy when compared to children born in other seasons. In one study, vitamin D insufficiency in infants was related to a 11-fold higher risk of developing peanut allergy and a 10-fold higher risk of developing multiple food allergies. Still, this finding is really only valid for children with parents born in Australia rather than those who had parents who migrated to Australia. And studies also show an association with prolonged vitamin D levels and IgE-mediated food allergy development, rather than just having kind of low vitamin D at a certain point in time. This consequently suggests that many vitamin D statuses are needed in early life to really impact immunity long term. Now, there are other risk factors that also increase the risk of developing food allergy, and these include things like parent atopy. So if parents have atopic conditions such as asthma or eczema or food allergy, and if children also have different atopic conditions, increasing gestational age, pet ownership, antibiotic use, and gut health such as the microbiome. And if you would like me to discuss these other risk factors in a bit more detail, then please do leave a review and contact me on various social media platforms that I will provide in the show notes. And I will be very, very happy to discuss these as well. Just let me know. And that's a wrap, guys. So thank you so much for listening. Please do leave a review and share this podcast with friends and family. You can also follow me on social media, which will be provided in the show notes, as well as everything that was discussed today, with citations if appropriate, of course. Now, until next time, thank you so much for listening. Bye-bye now. Now, just a disclaimer that the information in this podcast is for information purposes only and is not intended to be used as medical advice. I am not a medical doctor, so I have never and will never give medical advice. You should always speak to a healthcare provider about your unique health needs. My opinions are entirely my own and do not reflect the organisation's opinions that I work for. Due to the nature of my work, I can sometimes have access to unpublished literature. However, due to confidentiality, I will strictly not be discussing unpublished literature. I only discuss findings from published literature in this podcast. I am not responsible for any claims related to procedures, professionals, products, or methods discussed in this podcast, and I do not approve or endorse any products, professionals, services, or methods that may be discussed in this podcast. This podcast was written and produced by me, Ayah. Music was also produced by me, Ayah, using GarageBand.